Imagine a player moving through a complex three-dimensional corridor, tracking enemies, navigating obstacles, and making split-second decisions to survive a fast-paced shooter. This performance comes from a biological computer built by Cortical Labs, 200,000 living human neurons grown on a microelectrode array. This system, called the CL1, learns to play by translating game data into electrical pulses that the neurons then process to generate in-game actions. For decades, the baseline assumption in biology has been that complex learning and memory require a brain. We operate under the dogma that intelligence is a specialized product of high-level neurological hardware. However, that assumption is being challenged. Laboratories are documenting goal-directed behavior and associative learning in biological systems that possess absolutely zero neurons. Associative learning is the process of linking two different stimuli together, such as a dog learning to associate a bell with food. If biological brains are not the sole creators of cognition, then our understanding of how intelligence evolved is incomplete. We have to look past the skull to find the true origin of the mind. To find that origin, we must move down the biological scale to the microscopic level. In February 2026, researchers at Harvard University published a study on the single-celled organism Stentor caeruleus. Stentor is a trumpet-shaped protozoan that lives in fresh water. Researchers paired a weak tap with a disruptive strong tap 1 second later, triggering the cell to contract into a ball. After repeated trials, the cell anticipated the stronger stimulus, contracting fully to the weak tap alone, predicting danger before it arrived. It has no nervous system, possesses no synapses, and has no brain. Yet, it successfully executed Pavlovian conditioning, a form of learning once thought to be exclusive to complex multicellular animals. This suggests that memory is not an evolutionary innovation of the brain, but a capability present at the very floor of biological organization. We can go even deeper, down to the molecular level. At Tufts University, Michael Levin's team studied gene regulatory networks, or GRNs. GRNs are the internal molecular circuits that cells use to make decisions about which genes to activate. When these molecular networks were trained using associative conditioning, they didn't just learn a new response, they physically reorganized their causal structure. They became more unified and coherent, behaving more like a singular, purposeful agent. But this capacity isn't limited to biology. In 2024, researchers at the University of Reading conducted an experiment using polymer hydrogels. A polymer hydrogel is a synthetic, jelly-like material that can conduct electricity through the movement of ions. They plugged this synthetic jelly into a retro video game and provided it with electrical feedback. Despite having no biological components, the hydrogel learned to play. As the game sent pulses through the material, the ions within the gel shifted, clustering and rerouting their pathways in response to the feedback. This spatial distribution of ions created an emergent memory that allowed the jelly to improve its accuracy. Whether we look at synthetic polymers, single cells, or human neurons, the same adaptive mechanics are at work. The evidence shows that cognition and memory are substrate-independent, meaning they can manifest in vastly different types of matter, as long as that matter is sufficiently organized. To explain these findings, the volitional agent criterion framework suggests that agency, the ability to act with purpose, is an intrinsic property of complex organization itself. The hydrogel experiment proves that memory can be encoded simply through the way charged particles distribute themselves in space. This suggests that memory is not a biological invention, but a physical property of how information is organized within a three-dimensional volume. This points to the space memory hypothesis, the concept that the quantum fabric of reality itself retains information through its structural geometry. Under this view, the vacuum of space functions much like the hydrogel's matrix, holding structural impressions of the energy that passes through it. This implies that biological memory systems are not generating information from nothing. Instead, they may be specialized interfaces, reading and writing to a memory field that is already built into the architecture of space-time. If memory is a property of space, and even single cells can learn, we have to rethink the role of our most complex organ. The brain did not invent the mind. It is a biological amplifier, evolved to scale up and integrate cognitive properties that have existed since the dawn of the universe. In this framework, neurons are not the source of intelligence. They are the tools nature used to bring it to its highest possible resolution.